All right. Good afternoon, everyone. Please join me in welcoming Dr. Coyote Jagade, Assistant Professor of Environmental Toxicology at UC Davis. Dr. Jagade's research explores the intersection of pollution and ecosystem health, with a particular focus on terrestrial ecosystems. By studying microarthropods and other soil-based invertebrates as bioindicators, his work sheds light on how environmental contaminants impact soil health and broader ecological function, specifically pollutants associated with agriculture, such as pesticides, metals, PFAS, plastics, nutrients, and pharmaceuticals. His work examines how pollution affects soil organisms, ecosystems, and ultimately the health of animals and humans. Dr. Jagade earned his PhD in toxicology from the University of Saskatchewan in 2019 and holds a master's in zoology from Obafemi Awolowo University of Nigeria. He joined the Department of Environmental Toxicology, I believe, in 2023, 2024. And his talk today is Advancing Realistic Chemical Risk Assessment, Evaluating Relevant Mixture and Soil Test Species for Environmental Protection. Yeah.
Well, thank you, Christina, for that talk. Can you please hear me? Can you hear me? All right, yeah. Thank you very much for that introduction. So I'm Coyote Jagade, as we all know. And I work here in UC Davis. I work in UC Davis as a cooperative extension specialist and agricultural toxicologist. So just like Christina has read, I'm interested in a lot of contaminants, any contaminants that has to do with agriculture. So we're talking of plastics now, we're talking of PFAS, we're talking of pesticides, metals, and pharmaceutical. So, and I have a joint appointment with the School of Veterinary Medicine, where I also work 50% appointment with the School of Veterinary Medicine and 50% appointment with the Environmental Toxicology under the College of Agriculture and Environmental Sciences. So today I'll be talking about soil. I'm sure, we've had some discussions today that, most of you guys are more into the marine and water and aquatic and all. But today I'll just be bringing something different. So I'm talking about soil. And of course, there'll be some intersection somewhere, in terms of research. So today I'll be talking about soil, as I said. And then one thing about this talk is I always try to generate data for risk assessment. But then I always look at how can we generate better data or data that we can be more confident about. Like, so for example, when we do risk assessment, most of the times we focus on the single toxicants or single contaminants, whereas in nature, realistically, you have mixtures. So that's one area I'm very interested in. And secondly, we test with species. So species that we use are actually very limited. But then I'm also interested in expanding, the retinue of species that we can use for tests. So this talk will be based on those two, species testing, new species, and also mixture toxicity. All right, let's delve into it. So, one movie. So it's okay.
Nope, maybe you have to go.
Fine.
Yeah. There you go.
So can I use this?
I think it now should work normally. Now
Okay,
it should.
Okay, all right. Yeah, so today I'll be starting with what soil is not. So people just take soil as dirt, like it's just dirt, doesn't make sense. We don't use it for anything, but it's not just dirt. So soil is not simple. It can be very, very complex. And it's not straightforward. That's the reason. So it's not fully understood because of that. And soil fertility is not permanent. So some people might just think we can keep planting, we can keep using the soil, the fertility is still there, the organic matter is there, but organic matter can be degraded. and you can lose your soil. In fact, there are some soils we call dead soils. They are dead, and they were formerly living soils, but they are dead because they can't perform the functions anymore. So when we talk about soil, the fertility is not permanent. So soil can actually be destroyed in a way. So that's what soil is not. And then, like I said, it's called dirt by many people, but then there's a definition for soil. So I prefer to use the third one here, which says soil is biologically active. So soil is like living. So we don't usually call in soil science, we don't just take soil as just a medium. We take soil as a living entity, that is developed in the uppermost layer of the earth crust. And then why is it living? So the reason why we say soil is living is because it has biodiversity that makes it function. So a soil won't function without the organisms, without the life in it. So because the life is intertwined into the soil, we say the soil is living, but then the living entity of the soil is based on those species that live in the soil. And so those species are the ones that make the soil do whatever it wants to do. And then when we talk about soil health from a biological point of view, there are different definitions. So for us, soil health will be ability of the soil to continually produce the ecosystem services, to provide ecosystem services for us or for the environment. So especially supporting biodiversity. And then if we look at this is the ecosystem service. These are some of the ecosystem services that can be provided by soil. So as you can see, you have nutrient cycling, you have so many things here. And then soil provides us with so many things. For example, we all know soil provides us with food, with pharmaceuticals, raw materials for pharmaceuticals and all that. So that's a brief about soil. I won't go more into that. But then because of the importance of soil, we tend to protect soils. And that's where my work comes in. So as a soil ecotoxicologist, I try to look at how to protect soils, how to make sure soil continues to provide these ecosystem services. So one way to do that very quickly will be through soil ecotoxicology. So we know soil, there are different threats to soil, but then I'm coming from the aspect of contamination, of chemicals, of metals, or whatever. So and that's where I'll be introducing the term soil ecotoxicology. So what does ecotoxicology mean? Maybe we all understand it, but for soil ecotoxicology is just looking at how toxins or contaminants affect the ecology, within the soil. And we know once the ecology is affected, then your soil health is affected, and the soil is impaired, right? So, but for soil ecotoxicology, because of these issues, there are tests that have been developed, to investigate problems with soil, and then in order for us to protect soil. So the first set of tests were done because of pesticides, it was during the time of the silent spring and all that, and then they felt that, these soils are not producing, they found that earthworms were dying and all that, and then they started doing some tests, and by some, foremost scientists here, like Edwards, here in the United States, they started this field of science, soil ecotoxicology. And then after some time, we started developing protocols, like the OECD developed guidelines for ecotoxicology testing. So we have guidelines that are developed for earthworms, for springtails that we use. So we follow those protocols, and then we use them as tests. Of course, we modify those protocols depending on the question we want to answer, right? But then we follow this strictly. Especially if you are trying to regulate, if you are registering pesticides, you have to do this test. For any registration of pesticides, you have to do the soil ecotoxicology test. So it has to pass the test. It has to be, you have to be able to convince the regulators that it's not toxic. It won't kill all the organisms, right? So, and then, like I said initially, I'm focused on test species. So because that's very important for you to do any ecotoxicology test and for you to get the right results. So we have some standardized species, and then there are conditions for using the species. The reason why they are standardized because they can be universally used, and they are easy to actually use for testing. So these are some of the conditions. They are ecologically relevant. easily bred in the lab. They are sensitive and well distributed in the environment. So, and for me, I'm specialized in soil invertebrates. So I look at microarthropods and annelids, especially. I've also worked with snails, mollusks, but then my main focus has always been with microarthropods and then earthworms. So I'll be introducing some of my species to you. So here we have the predatory mites. So I worked extensively on these as well. So they are usually used in biocontrol, for biocontrol when you are dealing with IPMs. So they destroy other pests, and they are very good bioindicators of contamination in the environment. So there are protocols for this, standardized OECD protocols for this. This is the Opia nitens, which is the Oribatid mite. That's the first Oribatid mite that's standardized for toxic testing. That's the Opia nitens, and it was just standardized in 2020. So we have the protocol for that. These are earthworms. So these are Eisenia foetida, which are the standardized earthworms for test, foetida or Andre, so any of the species. So we use them as standardized species. And these are enchytrates. So that will be Enchytros crypticus. So these are also annelids, but they are smaller, they are very small, tiny white worms. And they are also standard species for test. And then we have, lastly, the Collembola springtails, which is the Phosomia candida. These are species that we use. However, there is always this issue with these species. This is a compost worm. So you find them in very high organic soils, not generally in soil. So for me, the question I ask is it actually representative of soils? So these are questions I'm asking of these test species. And also Phosomia candida as well. You find them almost everywhere, on soil surfaces. They are also found mostly in very high organic soils. So they may not be as representative as we thought they are. And that's why I try to look at other species, maybe local species that we can also use. But then my criteria for using the local species will be, can we easily breed them in the lab like this? Once we can satisfy that, then I try to go into that. So this one actually passed the test because this is a true soil species, and was developed by it was a project spearheaded by Environment Canada. I was also involved in the development of this protocol. So it's the first main true soil species, and based on that, that has also inspired me to do more to see if I can culture or get more native true soil species to use for toxic testing. Let me go on. So these are some of the guidelines that we have for these species that I mentioned. And then I'll be going to one of the research I did with the Opia nitens. So this was a study we did in Canada. So it was about the Opia nitens. But before I go into Opia nitens, I want to discuss about Oribatid mites. So one of the things that you can see, Oribatid mites have been around for about 350 million years. So they are pretty well developed. They've evolved over time. But then we just have the first one in 2020 as standardized species compared to the others. And that's why I'm very interested in standardizing more Oribatid mites, because we know they are well evolved and they actually represent the environment. So I just wanted to chip in some of the some of the information about Oribatid mites here. So this is the Opia nitens. Like I said, it was the only standardized Oribatid mite for toxic test. So let me go on to another study. Yes, so this is the study I really want to actually start with. So I'll give you a little bit of background about this. So this study was, the reason why we had this, we're trying to use a local mite species that is related to tropics. Because in Nigeria, for example, we import 100% of the pesticides used in Nigeria imported. So that's not the problem. But the problem is that we usually follow the manufacturer's recommended use, right? The recommended dosage, right? So, but then the problem we now have is, right, we are following the manufacturer's recommended usage, but then the tests that they use are from the template, because most of these pesticides are, they are manufactured maybe in the US, in Canada, in Europe, and all that. And these are temperate regions. So most of these tests are based on 20 degrees Celsius. We all know that. They are based on those standardized species, which are temperate species. And then we now began to ask the question that, okay, why don't we use this for our own local species, right? So, and then we started looking for what local species can we use? And then after investigation, we found molecular species to be appropriate for those kind of tests. We actually had a lot of mite species, but many of them died. We couldn't culture them in the lab. But for some reason, these species actually did well in the lab. I don't have the real picture of this. This is just the, I don't have the colored picture for this. But these species did well in the lab, we were able to reproduce them in the lab over time. And then we thought we could use them for tests. So we decided to, to design tests for this species. So the idea is that we want to test these pesticides on our own local species using our own local conditions. For example, temperature. Instead of using 20, we rather use maybe 26 or 27 or 28, which is representative of the tropics. So that's a little bit of the background to this. And then I'll go on to show you what we did. So before we use this species for toxicity testing, we had to optimize. So we already have the information about the standardized species, from the OPR-nitens, the predatory mite, the earthworms, the collembola. And then we're like, okay, we don't just want to use the information we have. We want to see if this works for us. So for this, we try to optimize. We expose these mites we put them in different soil quantity, 5 grams, 20 grams, 40 grams, and then we observe them over time to see how they perform. And then also we also expose them to different temperatures at the same time. So 20, 22, like that, to see if they performed well. And then we expose them for 14, 21 days to see the optimized duration of time to get reproduction. So the long and short, I don't want to go into the whole paper, but the long and short is that we found that 20 grams was the optimized, the best quantity of soil for them to reproduce and perform very well. And then they did well at 28. So at 20 degrees, which is the temperate temperature, they didn't reproduce. They didn't do well at all. So, which means that it is right that we have to be using our own local species for testing. And then the best day is, so 28 days was actually the best duration, which is also the same with most of the standard species we use anyway. So, and then with this, we went to do the research. So this is the study. I'm not going into the methods because I have so many things to talk about today, but this is the paper that was published with about the molecular which we use. And then one thing I want to bring to your attention is the sensitivity. So for example, for cadmium, the EC50 was about 15 milligrams per kilogram. And compared to other mite species in literature, you see that they are very sensitive to cadmium. So, but for dimethoate, which is the pesticide, they are not the most sensitive, but they are quite almost the same with other standardized species. But then for this cadmium, this actually showed us that we need to be using these species for testing instead of using the temperate species. You can see that they are very sensitive. So if you are using temperate species in a country like Nigeria, then for cadmium, you are not protecting the environment. And that's what this actually shows. So I go on to the second study, and this is with the OPR-nitens, the one we did in Canada. So the first one was about test species. So this second one is about mixture toxicity. So although OPR-nitens is a relatively new species, but then I'll be focusing more on the metal mixture toxicity here. So like I said, risk assessment for me is based on being realistic. So using mixtures and also the test species is very important. So this is an example of mixture toxicity. So in this case, we're looking at mixture toxicity of 5 metals in Canadian soils. So these metals are usually found in mining sites. So this is like the distribution of the mining sites in Canada. So we got soils from these places all across Canada, right? So, and then we analyze the soil for these metals, for these 5 metals. So, and then we found that these metals are all there in the soils at elevated level, but then they were in the soils at particular ratios. So for example, if you go to a particular mine, maybe for example here, the copper, lead, zinc, and all that will be in this place at a particular ratio. They are quite different from one another. And then we designed a test. So we're trying to look at site-specific risk assessment. So instead of designing a test that will be general, we try to base it on the specific ratios of those metals. And that's using a fixed ratio method. So, and then we had about 10 different ratios of metals, and then as a fixed ratio rate, and then we increase them as a dosage, like as a dose response, in a dose response design. So we just increase this by maybe ×2 and all that. So this is just this fixed ratio rate design. And then we expose our OPR-90s. So these are, we expose them in the lab. So these are the test tube. They're under 20 degrees, 400 lux light, and 60% humidity. So these conditions are the protocol, are the standardized protocol for OPR-90s use. And then we extract them by heat. So quickly I can just explain. So how we extract them from soil, we just when they are in the soil, you apply heat or light source from the top, and then you have like a funnel where they drop down through like a mesh. And that's how we extract them, because these mites, the idea is that they try to run away from the heat, and then they jump down into the trap, like a trap. So we just get them and then we count. So, and then we perform the dose response test with that. So quickly to the result. So this is just showing the EC10s of these metals. But the more interesting one is for the mixtures, which I want to show you. Here nickel was the most toxic. This was the first time we tested nickel with OPR-90s, and we never knew it was the most toxic metal. So we're thinking it was copper or cadmium, but nickel was very, very toxic to them. That's just one example of some of the observations or new findings that we found. But then I'm interested in the mixture toxicity actually. So for the mixtures, you can see these are different EC10s that we found in this soil. But one thing, the most important thing is this. So what we found was that based on EC10, this line means concentration addition. So that means that the metals are not interacting. They're just additive. But then if you look at it, whenever these bars are not up to 1, when they are less than 1, it means synergism. So one thing we found is that out of about 10 different observations, we found about 70% were actually synergistic. So which means that it is important for us to always look at mixtures, not just singles. So because 70% here shows synergism, which means that we might not have been protecting the environment if we're just ordinarily, or if I just adding together, just using concentration addition, or just based on single metal. So this actually shows synergism of these metals, and then that made us know that we need to do mixture toxic test for risk assessment. Then in continuation of OPR-90 study, after we got that, we then decided to do genome sequencing of OPR-90. So we want to go further into genomics and the molecular biology of this. And so this is submitted now. And then one thing we found out was that the genome content of OPR-90 was actually smaller than most mites. And we don't know the reason. When we compared it with different other mites, they were very small. But one thing I know is that OP-NI10 is quite very tolerant. So we don't know if there are relationship between that and, and the genome content. But then that's what we found out, and we hope this will be accepted, and then we can have it out there. So the next study I'll be looking at will be mixture toxicity as well. But then that was the work we did in Europe, in the Netherlands. So with that, I wasn't using mites, but I was using earthworms. So I'll just briefly give you a background of the study. So for this study, it's a very big European project. And then we're looking at pesticide mixtures across different countries in Europe, right? So, and then we chose about 8 different cropping systems across Europe. And then we monitored for pesticide residue. So it was a very big study looking at pesticide residues. So, and then quickly to the result. So one thing we found is that when we looked at all the compounds, so this would be your conventional farm or integrated farm, and then this would be your organic farms. So you can see these are different pesticide residue mixtures. So red just means 1 compound detected. This yellow or so means 6 to 9 compound detected. So that's about 33% of all soils in Europe. About 6 to 9, at least pesticide residues were detected in the soil. Some cases we have 14 to 17 compounds detected. So which means, so this is just to establish that most of these pesticides are in the environment as mixtures. They are not just there single. So if we are trying to protect the environment and say, oh, we have glyphosate at safe levels, but then are you considering lambda cyhalothrin? Are you considering dimethoate? Are you considering the others? So this is just what we wanted to establish, that these things are there as, they are not just one, but they are many. And then this, and you can see the organic site as well. So about 24%, you have 6 to 9. These are organic farms. Then 2 to 5, almost 69%. But this is when you are considering all the compounds. But when we now delineated it to the compounds that are still being used. So this is all this contains both banned and both currently used pesticides. So but then when we now delineated it to the ones that are approved only, we see had about 40%, about 2 to 5 compounds detected in 40% of sites, and then 6 to 9 compounds detected in about 27% of sites. So, and then we also have the same with the organic compound. After taking out your DDTs and DDEs and other banned pesticides. So, and then this made us realize that if we do any test, it has to be mixture toxicity testing. It has to be mixtures. We can't just do one single pesticide test and then we think we are protecting the environment. So in order to do this, it can be very complex if you want to do a toxic testing for about 10 pesticides at once in the lab. So one thing we now did was to prioritize. So, and then I was tasked with the assignment to prioritize this pesticide for use. So then they were like, okay, because we found that many of these pesticides are about 2 to 5 compounds, then we said, okay, for most soils, we will prioritize 5 pesticides for mixture toxic testing. So these 5 pesticides will be based on the pesticides that we know will drive the toxicity, right? So this is the way we did that. So one thing we did was to look at the toxicity of the pesticides. So we went to the literature, went to the regulatory literature, and then just to check what are the toxicity values of these pesticides, of all the pesticides. For example, if you have 20 pesticides, you look at all the toxicity values, like the EC50s, the NOx, no observed effect, EC50s. And then after that, for your NOx, we applied like an assessment factor divided by 5, and for LC50 or EC50, we applied 10. And then we found the risk quotient based on your PEC, that's predicted environmental concentration. So we have these values also in the regulatory guidelines. So we are using EFSA which is the European Food Safety Authority. So they have all these numbers, and then we just use the PECs from that, and then divided by our PNEC to get our risk quotient. So the higher your risk quotient, the more, of course, the more risky that pesticide is. So that's one criteria to choose the pesticide. And then we have also added other criteria. So for example, we had to also look at the persistence. So if pesticide is persistent, we also scored it high, being that if it's persistent, it means it would still remain in the soil to cause initial toxicity. So that was also another criteria we used, persistence, and then that's using the DT50, that the degradation time, 50% degradation time as a persistent indicator. And then we also looked at the occurrence. So how much of this pesticide, what's the occurrence like? If you have like 20 soils, if let's say glyphosate now, if you have 20 soils, how frequent is glyphosate occurring in about 20 soils? So for example, glyphosate was actually one of the most occurring pesticides, glyphosate and Captan. So finally, like Captan was almost in 100% of the soils. Glyphosate was almost in 80–90% of the soils. So that was another criteria we used. And then we normalized it, just divided by the highest value there. We normalized it to about 1, and then we multiplied everything together. So, let me see. We had the answer there. Yeah, so we had a total score, for example. So we added, we normalized and then we multiplied this together to get the total score. So the higher the score, the higher the priority for the pesticide. And then as you can see, this was the highest, so pesticide C will be the highest, pesticide will be the most prioritized pesticide, followed by A, D, B. And that's how we chose the first 5 pesticides out of about 20 or 40. And those pesticides were now used for toxic testing. So we prioritized that. So we believe that these 5 pesticides are the ones that are truly driving toxicity. So yeah, and that's what we did. Let me see my time. Oh man. All right, so we also use this species. So one thing I want to say is this. This is an European local species. This is a true soil earthworm. And then for this study, we're trying to also look at using representative species. Like I told you initially, this is a compost worm. It's not truly representative. You only find them mostly in organic soils. But then these are found in most soils, organic or not organic, just general soils, you find this there. So we decided to also use this as part of the test, just to make sure we are realistic to an extent. And then to also compare how sensitive these native species are to the standardized species. So very quickly to the results. This is for Fetida. So one thing I just want to point out is all these means that they are significantly different from the controls. But so for this test, very quickly, I have a lot to say here. So for the controls, no pesticide. For the MEC means the measured environmental concentration. So these were based on the pesticide we found in the field, based on the concentrations we found in the field, and then we dosed that in the lab. And then for the PEC, these are based on the PEC concentrations, the ones we found, we got from the EFSA database. So we dose the pesticide based on the pesticide predicted environmental concentration, and then to get a higher dose, we multiply this by 5, just to kind of do something like a dose response analysis in the lab, right? And then, but one thing we found that for this species, most of the effect were at 5 pec, right? So they were at 5 pec. Although at 5 pec, they were at 5 pec, but they were mixture toxicity. So because at 5 pec, most of these pesticides, they were lower than the EC50s. The pesticides were actually very low, the low concentrations. But when at 5 pec, and then when they are consuming mixtures, they became more toxic. So we found effect at 5 pec more than the others. But it was quite safe because at pec, we didn't find so much effect. So to an extent, we were like, okay, it's still fine. But there are cases where you might overuse pesticides, maybe accidentally, and then you will be having this kind of effect. So And then these are so these are countries, these are abbreviation of countries. So we only found effect in about 5 countries with this species. And then for let me quickly go to this. For this, we also found, so for this Lubricus rubellus, this is a native species. We also found effect, but mostly at 5 peg as well, just like the standard species. But the difference is that in some countries where the standard species were not sensitive, the native species were sensitive. So that was the difference. So that was like an added bonus for us to for the protection of the environment. So for example, if we are just stayed with the standard species and say, wow, standard species we're affected in just 5 countries, then we are fine. But then if you now use native species, that increase our detection, the rate of detection of how risky the sites were. Very quickly to Phosomia candida. For Phosomia candida, we found effect at peg concentrations in some cases. And these were because of the mixtures. So for these pesticides, all these pesticides, like I said, they were all low concentration. So if we're just considering them singly, we won't find any effect. But because they were in mixtures, even at peg, we found some effect like this. At peg concentrations, we found considerably some significant effect with the Phosomia candida. And then, like I said, we were able to increase our detection of risk. So if we are let me go back. So this is the standard species, for example. So this is standard species. With the standard species, you're only able to detect 5. But when we increased it when we added the native species, we're able to detect risk in 8 different countries. But then when we added the Phosomia candida, all the countries were actually banned. So that's to show that we need to increase the number of test species we need to use, and then also shows that we need to also include native species because we're able to detect more with the native species. Yeah, so these are just the conclusions for that. So one thing I want to bring, native species should be increasingly used as bioindicators. And then we should also consider pesticide mixtures in risk assessment. So this is another study I did. Yes, so this is another study I did in Netherlands. So apart from just taking your soil and putting your pesticides in them, I was now trying to look at realistic toxicity now. So how do we look at spray? So for those ones we didn't spray, but these ones were spraying based on pesticide spray schedule. For example, maybe for corn, for a corn farmer, there are some pesticides that you have to use at some certain times. So you have to apply, maybe you have to apply glyphosate first 1 week, then after 2 weeks, apply this and that. So we follow the exact scheme, right? And then the only difference is that we did that in the lab. So we're spraying just like you apply, we apply pesticide by spraying, right? So we also sprayed on the soil surface. We didn't mix with the soil like we normally do, just to see the effects, just to see how we can bring the field into the lab, kind of. That's what we're trying to do here. So, but for this test, I decided to go only with the native species, just to see what will happen. So this is the Lubricus rubellus, and this is also another native species. So they call this the night crawler. So it's a very bigger earthworm, and then it usually lives down in the soil. So they call them anesic worms because they live down in the soil. They only come to the surface at night to browse through the organic matter, litter, and then go back down. They are very big. But these guys are epigeic worms, so they are always on the surface, just like the standard species. And so, and then again, one other thing is that we also modified the test for this because they are larger. We used, more quantity of soil, about 1.8 kilograms of soil, And then we had a different setup for them, like a glass plate. So for this experiment, apart from just looking at survival and reproduction, we also try to look at their burrowing rate. So this setup made us to also observe that how they burrow into the soil, their burrowing rate, did it decrease or increase because of the pesticide? That is, we are looking at ecosystem services that they provide. And also this also helped us to quantify the casts. So that shows their nutrient cycling, because their cast is very nutrient rich. You know, it's just like they take the soil, they heat the organic matter, and then they form these casts, which are like fertilizers, right? So, and then we also used this setup we were able to use this setup to also analyze that. So very quickly, this is the result. I just want to show you this. If you can see the controls, we had reproduction in the controls, but when we sprayed, all of them did not reproduce except one, except Czech Republic. So there was no reproduction at all. So which means that the pesticides were actually very toxic to them. So that's for the spray, that's for the rubellus. And then, although we found some effect on survival with the Netherlands, but then this was the most glaring effect that we saw. So for this, growth was also affected. Survival for Portugal was really affected. Yeah, most of them died with the spray. They also had some effect with the cast production. So for Portugal, they didn't produce enough casts, which means that their nutrient cycling was impaired. Nutrient cycling function was impaired. And then if you see the growth, you can see the effect on growth. So they didn't grow well. Many of them had very stunted, impaired growth. So they reduced in size and weight, which means the pesticides was affecting them. And then, so this was just to show that if we do our research realistically, we can know what really happens in the field, probably. Because we just copied the field and just brought to the lab, and we are seeing this effect in real time. And these are very low concentration of pesticides. But because they are in mixtures and because they are in spray series over time, that's why we are seeing this effect. So this is like a model of the degradation rate of this pesticide. So one thing I want you to pick out from here is that Netherlands and Portugal, they actually had the highest residue over time. And not only that, they had the most number of pesticides. So active substance about 10, and then for Portugal, 10. And then the result also shows this, because the results show that these two countries had the most effect on the earthworms. Maybe because of course the number of pesticides, and then pesticides were persistent over time. So they didn't degrade like the others, they were persistent over time. As you can see. So if you can see at the lowest level, about 0.0015 milligrams per kilogram. And then also almost the same thing with Netherlands. Now, if you look at Czech, for example, at the lowest level is about 0.0005. And that's why we didn't see so much effect. So that this is based on degradation rates. We just modeled it just to show degradation of pesticides and then adding them together. So we can see why these two caused most effect. So in conclusion, cumulative pesticide residue levels were highest for, like I said, and then the earthworms were impacted differently by the pesticides. So different results based on the earthworms as well. So this also means that we should use more species. Like these species have different responses. Netherlands impacted this more, while Portugal impacted this more. And then to the Californian project. So one thing I'm trying to do very quickly, this is just about last 5 minutes, and then we can take questions. So I was talking with the students today. I was working, so I work with the vineyard growers, and then wanted to do some sampling in their soil just to start working on soil health and how I can help them better. So there's a background to that. Last year, there was some contamination in these soils, and then they had difficulty selling their produce. So most buyers didn't buy. They reached out to me to help them look at the metal concentration and all that. I did that. From my assessment, they were fine. Although they were elevated levels, but they were fine. But the buyers anyway said they were not buying. They were not ready to risk anything, So of course, they lost a lot of money because of that. And then that got me thinking, like, okay, let me actually do more research with these people. Let me know the state of their soil health, and then it will be easy for me to profile solutions. So for example, if a buyer comes and then I have studies done already, to be easy for me to say, well, we studied them, the soil is good and all that. But if we don't have anything, there's nothing we can tell the buyer. And then the buyer might say, well, I don't want to buy, I don't trust your product. So this made me thinking. And then for this, we went out, I had this student working with me from Brazil. So we went out collecting soils from different buyers across California. And then one thing I wanted to do was to assess their habitat quality, the quality of the soils. But the most important thing for me was to actually collect soil species native or native to California or representative of California. That was actually the goal of this project. And then I actually found some, as you can see here. So these are some of the species we found in the soil after extracting. So these are some, hypograssural species, collembolans. So we had more collembolans than mites in these soils, a lot of collembolans. And then what I did was that, like I said, my criteria is to see, can I culture these things in the lab, right? And one thing I do is that I feed them with yeast. So the standard species are usually fed with yeast, for the microarthropods. And then I said, okay, let me feed these guys with yeast as well. If any one of them survives, then that will be the species. If it doesn't survive, then that's it. So for many of the springtails, they actually survived in the lab. In fact, we are having so many cultures now. They are reproducing a lot. They are doing very well in the lab. And then my plan is to be using them for toxic tests going forward. And then I'll be doing that for mostly Californian soil. So with that, I'll be sure that whatever test I'm doing with them, that test is representative of Californian soil. Rather than just using one standard species that may not really work in the soils. So that's the idea. And we also found some mites. Most of the mites actually died, but there are some that were able to feed on the yeast, and they are also reproducing very well. So we have very healthy cultures of soils of microarthropods we collected from soils in California, and we're looking forward to using them for tests later this year. So that's what I've done so far. And then the next step is to optimize them. So just like I did for the mite species in Nigeria, in the tropics. So I wanted to see, so I want to optimize them for their duration. What is the right duration for them to reproduce? And then the soil quantity needed. But then for temperature, we don't need to do that because we count this place as temperate. So we'll just stay with 20 degrees Celsius as we have the lab. And then we keep them in the climate chamber. I have a climate chamber where I keep them. So at steady temperature and at 60% humidity. Then so far they are doing so well. So we hope to use them for tests later this year or in the summer. So, and that brings me to the end of my presentation. So far, this has been the affiliations I've had over the years. I've traveled far and wide, from Nigeria, Netherlands, Canada, and then California, UCNR. Thank you for listening. And I leave you with this quote. Thank you. Oh, okay.
So, seems like that trying to figure out which pesticides to mix together and to use for toxicity testing gets pretty complicated with a lot of potential pesticides to work with.
Yeah.
How does you balance like the increasing complexity of experiments when trying to manage which pesticides to use and where the cutoff is? And then is there potential to also incorporate other non-stressors like climate change?
Yeah. Yeah, thank you for that. That's a very good question. Yes, that's you can do that, but like you said, pesticide mixtures itself is complicated. So for now, we don't have a lot of data on that. So for now, we are trying to focus on that, generate as much data as possible, and then we can add other stressors. But I don't think it matters. If one wants to do it, you can always do it. It's just that it's a lot of tests, it's a lot of, it's a lot of work, but it's done. It's possible. And I think that's the right way to do it, actually, to look at external factors and all that. I did that with just single species, single chemicals. So I've looked at different temperatures, how they affect them, humidity, how they affect them. But then I've not done that for mixtures, which I think makes sense to do, just to see how, for example, climate change affects these things. So it's very important. And I agree with you, it's complex. It's a lot of work, but it's doable.
Yeah, I noticed in, you're comparing the survival growth, all that. Trees, that in Argentina for the tunneling and the tasks for the higher than people. I was wondering if you had any answer.
Yeah, good question. So that's one of the nuances of toxicity testing. So it could be hormetic effect. We don't know. Probably it is, because the pesticides were not, like I said, they were very low concentration pesticides, and a lot of toxic testing, if you do a lot of them, dose response, there are some hormetic effects at low concentrations. So it's more like the organism senses that there's a problem, and then they respond by, maybe reproducing more or produce, So that happens, and we felt that's what's happening there. So probably just, yeah.
I think it's like a stress response thing.
Yes, especially low level exposures. It happens a lot. Thanks.
So if I understood correctly, the spray method was actually much more toxic than your original method, which was actually mixing it in the soil.
Yes.
Which to me seems a little counterintuitive, but also concerning because that's the more ecologically relevant one. Do you have
Yes.
any ideas like why it's more toxic that way?
Yeah, I have an idea. So, when we mix, the soil, when we feed them, we put it on the soil surface, right? So there's a possibility that the food is not contaminated. So when we mix the soil, we now had food. We don't mix the soil with the food. We mix soil and hard food. But in this case of spraying, the food is on the surface and then we spray. So they are probably getting it from the food, So that's one. That's what we think. And then again, for the mixing, we usually use active ingredients. So we don't use the formulations. But for the spray, we actually use formulation. That's another thing. That's another added thing. So we use formulation, we use the ones that people use in the farms. So not the active stuff. So that could also, sometimes some of these formulations could have mixtures, and then
Do you think that's contributing to the toxicity more, or is it the method of application or a combination?
For me, I think that's very hard. So some studies have shown that some formulations actually are more toxic than the active because of the vehicle they use, So they say they use inert substance, to carry the pesticides. But then some of these substances can also be toxic. But That's some stuff. Generally, it's not the case. So for me, I would think it's the method of application. I would strongly say it's the method of application. So they were exposed to the food because we are spraying on the surface. So that's what I think. But the formulation could also be contributing to that. But since we didn't go deep into that, we won't be able to say.
Just one more follow-up, is that the standard way that people, do these experiments, is by the mixing method and not the more
Yes.
relevant one?
Yeah.
That's a very close finding then.
Yeah, that's how we do it. Yeah. It's very hard to do this because we have to follow the spray schedule. So some of these experiments took 8 months, 9 months, whereas most of the ones we do in the lab are usually 28 days, 56 days at most. But these ones were just based on how it was used in the year. We just followed that sequence. So yeah, so we applied more because of that. Okay.
You mentioned that you have the genome assembly for one of the mice. I was wondering, would that be used for toxicology tests, like looking at gene expression, or how would the genome be used?
Yeah, that's the idea. We want to use it for toxicology tests. So because that's the first time it's been done, we never had that. And then since it's a standard species, we felt we should have the genome assembly, and then we can use that for genetic expressions and toxicology. So it's basically, we want to use it for toxicology. But it's out there for anybody to use for any other thing. Then we are focused on toxicology. Yeah. Okay.
I actually have a big picture that your comment about soil being dead sometimes. Because that's something you've seen a lot, or is that something like rare that's more associated with extreme conditions?
Well, it's associated with extreme conditions. Yeah, but sometimes, for example, in Nigeria, like the Niger Delta region where we get our oil from, most of the soils there are dead. So people can't plant anything there. The soils are practically dead. And I'm talking of states, very large expanse of land. So, but it's because of spills over time. So it could happen. And we are just trying to tell people that these things could happen. Soils can be dead if you're not careful. So it's usually in extreme cases. Yeah. Okay. So you had a question. Okay. Yeah.
Okay. I mean, Dr. Chickadee for any talk.
Thank you for having me. Thank you. Done.